I'm here today to provide you some updates on biospecimens, highlights on numbers, and some principles of their existence in CBTN and as a shareable material. So obviously, we have not a trivial number of biospecimen in the center. And what comes to those, hope you can see those small fonts, but basically, in my view, we can divide the specimens into surgical specimen-related surgical specimens, as well as non-surgical specimens. And between the surgical ones are really, most of them are related to tissue. And the derivatives of those include frozen tissue, freezing media tissue, tumor models, which I'll be talking later on, primary cells, and obviously DNA or RNA nucleic acid from those. We also have surgical biofluids, which include cerebrospinal fluid and cyst fluid. We have also pellets from CSF, which I'm going to talk briefly about later. And on the non-surgical, we collect blood and saliva and all the derivatives coming from that. One additional specimen which I want to mention here is actually the adjacent normal tissue possibility, which one additional specimen which I want to mention here is really a pilot study which we're doing right now at CHOP, and it's only possible thanks to neurosurgeons teams, Jay Storm and his folks, who are trying to pilot and bring the adjacent so-called normal or corresponding tissue. It's obviously not a trivial thing, and it's not always possible, but and I guess in many centers this is already utilized. However, and the numbers are still small, but we all know the biological meaning of having non-tumor tissue for many of the biological question studies. So what about those 64,000 samples numbers? And obviously, not surprisingly, the first what comes is the DNA derivative from all of the specimens that include tumors, saliva, whole blood, and paternal and maternal material. Second comes to it is really a flash frozen tumor. And 11,000 sounds really a lot. So we were trying to really get to know what 11,000 really means. And this came up with exactly 2.1 kilogram of tissue within those vials. And it sounds a lot, and it also sounds a little, but if you have in perspective, adult tumor, teenager to adult tumor is really 1.5 kilogram, 1.4 kilogram around. So knowing that 11,000 aliquots of something so small and so light can take so much harm to those kids is something what I wanted to put into perspective. So obviously, the number of specimens, or maybe not obviously, but happily to announce that the number of specimens in Biobank are really increasing, and they stably increasing even during COVID. And apologize, the first one is actually frozen tissue. This is whole blood. This is a number of PBMCs, if I'm not mistaken. And lastly, this is CSF and plasma, which I'm really excited, especially because of my interest in a non-invasive aspect, which I'm going to talk later. And it all comes with an army of work. So briefly, just at the CHOP site, these are the numbers of processes which were made across CBTN for several projects, including the recent EXO one. And they're really impressive because each of these processes takes sometimes a whole day to complete. So the samples are really not here to only store. So I'm glad to provide that the numbers of samples delivered to researchers also growing along the years. And you can see this tremendous spike of especially tumor DNA and tumor RNA within 22 and 21. And that's clearly related to the EXO-1 project, which is really a highlight of recent tissue specimen-based studies in CBTN. So this brought me to This brought me to the aspect of checking what in which project used the most specimens and what are the top 5 projects within the CBTN to be used. And the fifth came CBTN 23, which is a proteomic project with over 300 aliquots shipped. And happily to announce almost 2 years ago, that paper came out from this work, but even more exciting part is the clinical trial, which started a few weeks ago, where part of that work really was meaningful for the that clinical aspect. So the second came CBTN 11, second and fourth actually, came CBTN 11, where there was close to 1,000 aliquots shipped between freezing media, specimens, cell lines, and as well as tumor DNA. And I found at least 2 papers already using that that specimens around ALT aspect in pediatric brain tumors and as a biorepository of our cell lines in the past. So the third in line is actually the methylation project. It's a relatively new project, and both data generation and analysis is ongoing. Not surprisingly, the second came project 12, and I tried to actually summarize how many manuscripts were submitted based on that project, but knowing that there's over 100 data projects in CBTN, I only highlighted the recent open PBTA collaboration between Childhood Cancer Data Lab from Alex Lemonade's stand and D3B, and basically an open approach where everyone was invited to analyze genomic data from that cohort. But I wouldn't have space on this slide really to include all of the papers which Project 12 and the first CBTN large data set affected. And lastly, as the spot is really EXO-1 with over 6,000 exact vials shipped for that project. And I was trying quickly to do a science, wondering how big impact that project would have. So assuming that the Roman X is 10 and adding 10 and 1, at least expecting 1,001 positive impacts in the future. But EXO-1, it actually brings me to something what brought me something to what Patty and Bev was just presenting, and I wanted to briefly highlight postmodern tissue with an EXO-1 data set. So not surprised to me, but close to a quarter of all of the tumor DNA shipped for that project were actually coming from postmodern tissue. And obviously, and unfortunately, not surprisingly, most of those were high-grade and diffuse midline glioma cases, but as well, medulloblastoma, ependymoma. This actually looked much better on 365 than on Google. So I really apologize for that. All right, so that's a large chunk, and that's something important. But I don't think I really catched at that point yet another aspect of that process. So within all of these postmodern cases, there's over 50 which actually have sampling on more than one tissue, tumor tissue fragment from each patient, and leaving those only orange ones. These are the cases where there is a huge possibility to explore spatial and temporal aspect of genomic information from these cases. And shifting briefly the gears for the liquid biopsy aspect, we actually wanted to share actually about pilot project which we started at CBTN, which not necessarily is that easy as CBTN is not saying that the collection at CBTN is easy, but we piloted biofluids collection protocol at CHOP, which actually focused on collecting not only the surgery cases for this biobank, but also the follow-up collections around MRI and therapeutic timeline for patients who visit CHOP. And we prioritize blood, plasma, and mononuclear cells from plasma for collection, as well as CSF. We also correct urine at surgery. I want to give shout out to Angela, not only here. Angela actually brought me to Gift from a Child program as well, but Angela put me in the position. What do we exactly want to do? What we want to do? How we want to do? And do we have timelines? Do we have budgets for that? When we came first time with this idea, and that was extremely, extremely helpful to kind of step back and think about this one more time. So briefly, I don't want to go too much over time, but we are the share specimens with few programs which are interested in this liquid biopsy questions. Anne from our group actually wanted to ask something around microRNA composition within those specimens, and we analyzed quite a cohort, and she specifically asked the question whether microRNA within plasma can be used as a monitoring tool. And we had only 17 samples around diffuse midline glioma and low-grade gliomas, which clustered together in the large cohort analysis. We took them separately. We had mean follow-up for 23 months of the clinical data. And what we found is that we had 3 clusters, and they're maybe not that impressive, but we're looking at another aspect of relapse. At the time of data analysis, we found that within first cluster, majority of low-grade gliomas, which often cluster together in DMG in our analysis, actually majority of those low-grade gliomas were already at the relapse state. Interestingly, these which did not were either concerning at MRI or they were already disseminated to the spine. And on the other cluster, that case we overseen before that it was collected during the chemotherapy, which might affect this significantly. So that actually got us really excited. We use differential expression analysis and machine learning, and this is just an example how relapse cases clustered significantly separated from not relapse cases in our study. And it's relatively early still. We analyzed independently those 2 free cases and comparing 2 free not relapsed cases with those which relapsed. And at that point, we collected blood at the resection and followed up those patients for 24 months. As you can see, looking at the top 15 microRNAs, there's only 5 presented here. We would be able to develop the algorithm of potential prediction of low-grade glioma progression from the plasma. And to this, it takes really an army. There's really hundreds of people who are involved in this, and I'm really thankful for their work. And thank you for listening.